hope everybody on there can hear me well. My name is Ashton Corbett, and today I'm here to present what I did over the summer. I worked on the AMR Genes project, so let's get into it. So a little bit before I can get into my actual presentation, I have to introduce y'all to what is an AMR gene. So AMR stands for antimicrobial resistance, and these are genes found in bacteria that basically allow it to develop a resistance to certain drugs that we use to be able to kill it, like methicillin, penicillin, various drugs like that. So, who cares? Well, there's been a growing debate over this, and you can clearly see how these drugs can be a growing problem within the United States and worldwide. For example, here we have MRSA, or methicillin-resistant Staphylococcus aureus, and it's actually resistant to all beta-lactam medications, including methicillin, oxacillin, penicillin, amoxicillin. All of those it's growing resistance to, and it's very pesky to deal with. So much, in fact, that you can see how it has risen through this graph. So MRSA, throughout the years in various different hospitals, has grown in cases in a linear path. And this graph was actually taken from the early 90s to about the mid-2000s. And you can see how the progression for hospitals has been increasing over this time period. I would imagine that if this graph was taken today, these numbers would be even higher. So it's becoming an increasing problem for not only our country, but worldwide. From this, now that I've given y'all a brief discussion about how AMR works, I can introduce y'all to what I did over the summer. Assuming that it would work. Okay, so here are the methods that I used for my project. So to start, I collected various water samples from really all around southwest Georgia. We got 3 different samples from the City of Omega's water treatment plant, 2 from the lakes in the front of the lab, the lagoon in the back, city and well water from the City of Omega, and a splash pool from Wild Adventures to test. So I'll be able to break down all of these. So first, when we brought the samples to the lab, we had to filter them down. So here's the filter I used. It's a 0.8 micrometer filter. And I basically used this pump to drain the water through the filter. And once it finished filtering, I was able to swab each of the filters and collect the samples and bring them to the DNA extraction room. So from here, I was able to add a lysis binding solution along with a lot of PBS to each of the samples that I collected. And this machine, the BioSprint, allows me to extract the DNA from my samples so that I can just get pure DNA to be able to use for my PCRs in the future. So for the PCR machine, I needed the plate. So for the plate, it's a 96 well plate with 3 negative controls and 6 positive controls. So really we're testing for about 87 genes with this plate. The plate already came with primers and probes in it, and this was it in the package. And in this box there's probably roughly 50. And then this was one that I already filled out and completed after I ran the test, just to show you the before and after. From here, once the plate was filled, I brought it to the PCR machine. So I would basically insert the plate into here, and I would use a microarray real-time PCR method. So for each cycle, here, I'll show you results. So for each cycle of the real-time PCR, my test would always run 40 cycles, and we're recording the fluorescence being shown from each of the wells. So during the PCR process, we know that the genes, or really the DNA, is being duplicated each time. So the first time you would start with 1 strand, and by cycle 2 you would have 2, and then by cycle 3 you'd have 4, 4 turns to 16, 256, and so forth. So by the end of the 40 cycle run, we really have millions of strands of DNA that allow us to see if the gene present in the well or not will be able to amplify through the graph. So if we get an early amplification, like in this example here on cycle 20, we know that that's a strong positive because it only took 20 cycles for the gene to amplify and cross over its threshold. Versus if a cycle were to occur later, so if there were to be a longer initiation phase and it were to enter into its exponential phase around, say, phase 30, that would be a weaker positive, but it would still be positive either way. So now that I've shown y'all how the graphs work, I can get to some of my results from my tests. So we'll first start with influent and effluent water samples. So this was basically the main course of what I did over the summer. So to start, we got 3 different samples which were taken within 2-week time periods. So we got 1 from the influent and 1 from the effluent, and both were filtered and swabbed and extracted and all that. here's some of the pictures of the lagoon itself and what I can show you. So on this side we have the inflow end, and this is basically all the raw water, bath water, water that's flushed down the toilet, sink water is all brought into here. It is traveled around this little curve here which collects it, and then this grate down here is used to filter the water and bring out all the things that have been flushed down the toilet, like underwear and such. All the big blobs are collected here and then they're disposed of, so that by the time we reach the pond, we're just left with pure water. Once we reach through here, we go into the big pond here, and the water trickles down through this pipe into here, and this is the oxidation pond. The oxidation pond is separated into 3 groups. It's kind of hard to see here, but there's these little black lines in the lake here that indicate the different sections of the pond. So essentially, the water passes through each section, and by the end of the pond, this is the start. The water should be oxidized enough to where it should be cleaned up. After it's been cleaned up, we move to the spray field here. So the water, we don't actually do anything with the water actually after it has been oxidized and treated. We just spray it on a field and then the grass is cut. So the water's not being introduced back into it. Well, it is being introduced back into the environment, but it's not being introduced back into humans or anything like that. Okay. So here are my results for the first run. On the left will always be the influent end, and on the right will always be the effluent end. So the influent end, as we can see here, has a ton of genes in it. There were roughly 25 genes within the influent end. These 2 up here on every graph, these are my positive controls. These just allow me to know that the test did work properly. But the main thing we're just trying to take away is that our influent end has a lot more genes than the effluent end, which is a good thing, which means that the oxidation pond did do its job of cleaning up the water a bit. Same thing here for run 2. So we can see a ton of genes over here versus very little, which is a good thing again. And likewise for run 3. So again, a lot of genes, very little genes. And now I can talk about my city and well water that I also was able to test. So for the city and well water, this is the city water. We collected a sample from the city of Omega, and that water is actually chlorinated and filtered to the highest degree. So we weren't expecting many positives to come out of that sample. And this well was actually right next to the oxidation pond, and we wanted to test this to see if there were any AMR genes in it that could have traveled over somehow. But both of these cases came back negative. So we knew that the city water was going to be a good negative, so we used this water as a negative control. And this one also came back negative, so I did not include the graphs for these because there was no amplification to show. Moving on to the Wild Adventures water. So it was taken from the splash pond. I'm sure a lot of y'all went swimming in this. And there were a few, but luckily there weren't many AMR genes in there, fortunately. So, but even though there were only a few, there still were some positives in there, so just keep that in mind. Moving on to the lake samples that I collected. These are just the positive controls. This is pretty much what a negative chart looks like. So we do have clean ducks. And moving on to the lagoon sample. So this is the lagoon in the back. This is where all of our wastewater for the lab goes. There are some AMR genes present within us, but there's quite less than the oxidation pond because the sample size is so much smaller. So for the lab, probably roughly about 50 people in here. And for the city of Omega's oxidation pond, they're treating probably about 1,500 people's wastewater that's coming in. So we expected there to be less genes, but they still are present within the sample. Moving on. Once I grabbed all of the data, I was able to compile it together. Let's see if I can pull that up. I made an Excel spreadsheet. With all of my data points.
Actually, can you just verify if the folks on the Zoom line can see that file?
Yeah. Can y'all see that?
They're not gonna be able to see it yet. But
Yes.
Share.
They can see. Awesome.
Alright, cool. Okay. So, like I said, the plate that I used tests for 87 genes. So I have all 87 of them listed out here. And this column right here, the B column, represents the family of drugs that the gene is immune against. And then I've labeled my sample, whether the specific gene was positive or negative in the sample, and then the period at which the gene crossed the threshold for it to be positive. So let's take the influent and effluent run too, because this is where my results were the strongest. We can see here how there's many positives within this case, and we have plenty of CT values to represent that, versus over here on the effluent end, we can see that there was way less positives. And compiling the data, we could also see that there were similar trends within the influent ends from the same samples. So if I were to click on, say, like column 4 here, and highlight it, we see that for the first influent sample, our CT value was 29, and our effluent sample, it was negative. So the oxidation pond did clean it up well. And that's similar to our results for the influent and effluent of the second run of the oxidation pond. So for the second run, we got a CT value of 27, and it was negative again, meaning that the oxidation pond had cleaned it up twice. From there, I was able to gather some genes that were really hot within each sample of the oxidation pond. So the genes I want to talk about are MEK, VEB, and OXA2. So MEK is responsible for resistance of all beta-lactam medications, and this is actually the gene that's found in MRSA that helps build its resistance to all of those medications. VEB and OXA2 are both found in E. coli, and both of these genes help for cephalosporins and other beta-lactam medications down there, such as oxacillin, which makes sense for these genes to be present in the water because it's gut, like bacteria is found in the gut of humans and such. So from there, I'm able to bring up to the conclusions, and the presence and growth of AMR genes is something to definitely be on the lookout for. They're growing, and there's a definite concern for them, and my project was aiming to target some of these genes and explain to y'all how they work and such. So here we are. Thanks. Y'all any questions? Do y'all have any questions?
Any question to Zoom land?
In the end, we couldn't see the slides because you got switched back and forth, so the share screen was not changed accordingly.
Did you see this?
What is this?
Right now, we're seeing the Excel file sheet.
Okay.
Maybe you want to click new share.
How do I share this?
Go back to
Go back to the Zoom, then press the green button,
Yes.
and then click your PowerPoint,
Okay.
then press share.
Okay. Can we see now?
Yes.
Okay. Were you able to hear me though?
Yeah. Yes.
Okay. So I'll just run through it one more time. So genes to know about: the MEK gene, the VEB gene, and the OXA2 gene. The MEK gene is in MRSA and is responsible for all the beta-lactam medications like penicillin and oxacillin. VEB and OXA2, those are found in E. coli, and those are responsible for its resistance to cephalosporins and other beta-lactam medications. And it makes sense for these to be in the oxidation pond because they're found in the gut bacteria of humans. So we know that the samples are corresponding to humans and how the wastewater has been treated. And some conclusions that I brought forth. So AMR is on the rise. It is very prevalent, and I hope that I was able to target some of these genes and expose them and shed some light on what's happening. So that's it. And do y'all have any questions now?
Ashton, I can start with, so, excellent work, first of all, and, this is not what we thought we will be starting when you came on week 1 to the lab, and we were looking at things to do the project. But and we have been using this kit for the last 3 years for different applications. So this is just one of the applications that we looked at. I didn't tell you anything about my question because I work with you, but what would you do beyond this knowledge? Like if you, because you are planning to be a physician and a cardiologist, and you have this tool in front of you, and there is no right or wrong answer, but what would be your next step based on this finding? Should we change anything with our oxidation process, improve it, or?
Yeah, great question. So if I would back up a few slides to the pond itself, so we can see here how the pond is covered in this green top layer, and this layer is actually a layer of duckweed, which actually isn't supposed to be over the pond. And I would imagine that if this layer weren't there and the sun were to actually able to properly oxidize the water, that the genes would be even cleaned up even more. So if the pond had been actually properly managed, I'd imagine that it would be even stronger, and there would probably be no genes in the effluent versus influent.
And the other follow-up is, like, if you look at the antimicrobial resistance genes in these samples, a project that is a future project in pipeline, and that would be looking for the microbial profile that we have in those samples. You are more than welcome to join us in fall. I am working with the PCOM, the College of Osteopathic Medicine. I have a team of DO doctors, students who will be working on looking at the zoonotic pathogens that might be present in these kinds of samples, because we don't have any data on what kind of bacteria, viruses are coming out of this. We would love to compare this data to see the consumption of antimicrobials in that community. So, there are lots of outcomes from your work that you have done, because it kind of show the resistance genes are against certain antibiotics. So what are the antibiotics most commonly used by the physicians in that area? And so we can actually kind of extrapolate the trends of prescriptions and kind of those kind of things. So you're very welcome to continue, and I'll let others ask questions if they have. Please, from the group. Anybody in the Zoom land?
Yeah, I have a question and also kind of like a comment.
Okay.
Do you know how does most of the antibiotic drug being developed?
I do not know how the antibiotic drugs are being developed. No, that's not something that I looked into during the summer.
I think my point is actually, so the origin of some of, most of the older antibiotic drugs that we use are actually originating naturally, that it was found, like maybe for example, penicillin is found in fungal species. They produce those antibiotic resistance proteins to help them survive, to help them adapt to the environment, etc. So this is antibiotic resistant gene is not necessarily all bad. It's human kind of discover the use to apply those antibiotic into drugs and into human medicine to advance our life. But my point is, sometimes it happens naturally, and some of the genes you found in the environment or from animals or from humans, they may not be all bad, so it's kind of like a balance of the life, the nature in our lives. Although I understand it is a hot topic, it is creating some of the resistance gene is is creating issues for human now. But when we deal with this topic, sometimes we need to take a step back to see, okay, are they really significant? Do they really mean something? Because some of the antibiotic resistant gene you can find them from animals that are not being treated by any antibiotics. They just happened because of the I guess the revolution. Like you need to adopt some of the infection from the antibiotic produced by the fungal species, and then the bacteria need to produce some proteins to kind of fight against it. So it's kind of, yeah, it's natural.
But Dr. Mosley brought a great point. It's like opening a can of worms. We don't know what we have, and that's like a Pandora box. But we have to draw a baseline. We have to know what we have in our community, and then we can answer a lot of these questions. And this could lead to a dozen of PhDs right here in South Georgia on exploring what we can do. We could be looking at the normal flora. We could be looking at the flora because the sample that we are looking at is from the environment. And it's also a way to check on the procedures that the cities take into account. This could be applied to the dairy farm effluents, because the dairy farm, we know it's a controlled area where we give antibiotics, and we know we want to monitor because those are that, we consume milk, we consume eggs, proteins. So it is good to have a baseline created, and this is like a kit that we use where we had 87 antimicrobial resistance genes looking at. There are thousands of genes, you cannot cover all of them. To do sequencing is very expensive. So this was kind of a dirty cheat sheet sort of a thing. Let's see what we have. And the good news is at least we feel better about the water that we are drinking, in that sense, swimming wise. But I agree with Dr. Mosley a lot. It's a nature's balance, and we need to explore this further. So agree that. Ashton, we have this certificate of completion for you, and we wish you all the best, and I hope you come back as a physician. He wants to be a cardiologist, and we need folks like you. So thank you, great job, great work, and wish you all the best.
Thank you.